So I'd like to take some time now and talk a little bit further about the details of independent assortment and how then we can do probability calculations on a Punnett square that's more than just the four possibilities when you have a monohybrid cross. So this is now what we call a dihybrid cross. So Mendel, looking at dihybrid crosses, developed his law of independent assortment. And so when you originally look at the P generation, you have a large yellow round P seed versus the second parent, which is a wrinkled green P seed. Now, two postulates can come from the crossing of these two parents. One, that you only find yellow and round, and you only find green and wrinkled. And so clearly the green, little y, and wrinkled, little r, could be packaged together. But you run the test, and what you find out is that they're not. So you get the possibility of finding green round P seeds as well as wrinkled yellow P seeds. So you get a phenotypic ratio of 9:3 to 3:1 with 4 different phenotypes now present. This is in essence like doing two Punnett squares at the same time. So each gamete that is produced from that F1 generation is a combination, is all the possible combinations of the parents, right? So you can get a big Y, big R, big Y, little r, little y, big R, little y, little r. And then you just line them up like you do in a standard Punnett square and then run out the genotypes of the resulting progeny. But the fact is here that he did see the two different characteristics that he was looking at, seed size or seed shape and seed color, that they independently assorted. So this is what we call the law of independent assortment, that each pair of alleles segregates independently of another pair of alleles. The opposite of independent assortment would be linked characteristics. Now we know this works because we understand chromosomes. He didn't. But we can look at the mechanism for this on a chromosome level. So the fact that we get random lining up of the pairs of homologous chromosomes on the metaphase plate dictates that you may or may not carry certain characteristics onto the progeny as groups or not. So let's say dad, which is blue, was round and yellow, and mom, which was red, is the green and wrinkled, right? And so if you wanted to keep round and yellow together, you need both the blue chromosomes, okay? If you want to keep green and wrinkled together, you need both the red chromosomes. But it's random, right? It doesn't. There's no propensity for one combination or the other, right? So this is where the flip of the coin comes in. So this is the law of independent assortment. Now, the limits on the law of independent assortment really kind of go back to that physical idea of loci on chromosomes. So it really only applies to genes on different non-homologous chromosomes. So genes that are located near each other do tend to be inherited together. These are called linked traits. Okay. Now, what you see then is that some traits are closer together than others. So in this case, you have the C gene, or the C locus, and the I locus. They are closer together than the I locus is to the G locus. So really what you're going to see is you're going to be able to measure that, and we'll talk about this later, but you'll be able to measure the distance between those based on how often you see a recombination event between those characteristics, okay, if they're on the same chromosome. So now, if you take that to the next level, you can actually get characteristics that are on the same chromosome to look like they sort independently. And that's because they're far enough away from each other that they almost guarantee a crossing over event. And then again, it's a flip of a coin, whether or not they're separated or not, just as if they were on separate chromosomes. So with that in mind, and taking our understanding of chromosomes and applying it to Mendel's work, you can really get a good understanding of how this works. Now, we can also just run the numbers, right? So we can apply multiplication and addition rules to predict the outcome of crosses involving multiple characteristics. So you can take a dihybrid or more trihybrid multi-character cross and basically do the math to get probabilities. And the best way to do that is just to work through an example. So remember, the first example, which should be getting intuitive for you if you look at enough Punnett squares. Sperm, are you going to get a big R or a little r? It's a 50-50. It's a toss of a coin. Eggs, big R, little r, and then you get the resulting Punnett square. But what if you had a more complex problem? What if you said, what are the chances that I'll get at least 2 recessive traits in the cross between what is listed above? Big P, big Y, little r, y, big R, little r, crossed with big P, little y, little r, little r. All right? So what you have here is 3 characteristics, 3 loci that you're looking at with 2 parents. In both parents, P is heterozygous. In the parent on the left, they're heterozygous for Y. The parent on the right is homozygous recessive for Y. The parent on the left is heterozygous for R. The parent on the right is homozygous for R. And this is the way you have to start to break it down. So the beauty of this is that you can break down then the probability of getting 2 recessive traits, right? So you list then all the combinations that you could get of recessive traits, add up the probability, right? So you're like doing a little Punnett square for each locus. So little p, what's the probability of little p coming out from a big P, little p cross? Well, it's 1/4. So you put 1/4 in there. What's the probability of getting a little y from a big Y, little y cross? 1/2, right? And so on and so forth. And you add all these up, you multiply them out rather, and then you add them up, and there's your chance at the end. This is pretty much plug and chug. Think about it. It will become apparent, and then we can practice this. All right? So keep that in mind.